Hi, my name is Jacob McPhail, and I'm an R&D scientist at Stressmark Biosciences, and I'm going to tell you today about our efforts in characterizing pathogenic protein aggregates for utilization as molecular tools to accelerate neurodegenerative disease research. It's quite the mouthful of a title. So we're based in Victoria, BC, on the west coast of Canada. We're not actually in Vancouver here. We're on Vancouver Island, so we're north of Seattle and west of Vancouver. There is a beautiful shot of our harbor there, and at Stressmark we produce life science reagents including proteins, conjugates, and immunoassays, and in recent years we've had a particular focus on pathological proteins involved in neurodegenerative diseases. So Alzheimer's and Parkinson's diseases are the most common neurodegenerative disorders. Alzheimer's is the 6th leading cause of death in the US, with recent estimates actually suggesting this is closer to 3rd in those aged 65 plus. This is becoming a greater issue as the population ages, as you could imagine, and despite the significant impacts on health and economics even, there are no cures known. So the key thing here is that both of these diseases are characterized by the presence of plaques in the brain, and these consist of protein aggregates which include the key proteins alpha-synuclein, tau, and amyloid beta, which I'm going to talk about to an extent today. So alpha-synuclein, tau, and A-beta share a common pathological mechanism, and this is going to be key to my talk today. So essentially these monomers are fairly flexible, freely soluble proteins, and during the pathology of this, these form these oligomers, and eventually these insoluble fibrils, which form the core of these plaques observed in these neurodegenerative diseases. And here at Stressmark, we seek to generate these different aggregated states in vitro so that we can use these as research tools in disease models and drug discovery. So talking about alpha-synuclein first, alpha-synuclein normally in the cell regulates synaptic vesicle trafficking in neurons. So here on the left you can see on a membrane it does have some alpha helical structure, but it also does have this flexible tail, and this is going to be important later in the talk when I talk about serine 129 and its phosphorylation, which is indicative of Parkinson's disease pathology. So alpha-synuclein, normally in solution, is an unfolded monomer, and it associates with these synaptic vesicle membranes to exert its functions. And essentially what happens, and there have been many different sort of inducers that have been studied of what causes this process, but what we know happens is these flexible monomers form these oligomers, as well as these large insoluble fibrils. So something causes this aggregation, and these proteins, pack on each other. And essentially, Parkinson's disease is characterized by alpha-synuclein aggregation into these toxic oligomers and fibrils. So here at Stressmark, we've generated several types of alpha-synuclein preformed fibrils in vitro. So these are recombinant proteins that we've been able to drive into preformed to study its effects in animal models. So there are several types of different fibrils related to the endotoxin level that's present, and also mutations. So familial Parkinson's A53T mutation that actually drives early onset Parkinson's, and these sort of more soluble filaments here. So looking at these alpha-synuclein preformed fibrils will actually seed monomers into fibrils. So monomers are those flexible states, and if you add some of these preformed relatively insoluble fibrils, it will actually induce further formation of fibrils. So here how we look at that is this thioflavin T molecule, when it binds to fibrils, its emission at 482 nanometers is increased. So on the right here is an alpha-synuclein seeding assay, and what you'll see in terms of just relative fluorescence units on the y-axis is over time these type 1 fibrils, which are formed with a little bit more endotoxin present, have a much larger signal and seed monomer and generate more of this THT signal a lot faster and to a greater degree over time versus type 2 fibrils, the line in the middle there. Despite there being, their presence is there under EM for both types of fibrils. So they are forming, but something is different in their structure that's affecting the thioflavin T binding. So taking these preformed fibrils and actually testing their effects in mice, first looking at the type 1 preformed fibrils, and these actually induce Parkinson's phosphoserine 129 pathology in a mouse brain. So remember I mentioned when I was talking about the alpha-synuclein molecule, it has that flexible tail that in Parkinson's disease becomes phosphorylated when it packs into these fibrils and these aggregates. So this is a single unilateral injection into the dorsal striatum 30 days prior. So one injection from one side. And what you'll see, monomer, which has the same level of endotoxin as the preformed fibrils, and you look at different concentrations here on the top, there's not really any signal for the phosphoserine 129. On the bottom row here, you'll see sort of this slight browning color on these mouse brain slices, indicating that phosphoserine 129 is now present after injection of these fibrils. And remember, because these are recombinant E. coli express proteins, the alpha-synuclein preformed fibrils are not phosphorylated prior to injection. So this is suggesting this is actually initiating phosphoserine 129 Parkinson's-related pathology in these mice. And essentially, here's a summary of several of these mice that these experiments were attempted on. And as you can see, this phosphoserine 129 pathology was not observed in any of the mice that were injected with just monomer. And in preformed fibrils at the lower concentration, most of the mice showed this pathology, and at the higher concentration, all of them did. So although there may be a small level of endotoxin present in the monomers and the preformed fibrils, this control showing monomer is indicating that the endotoxin isn't having the effect. It's actually the preformed fibril that's initiating that pathology. And looking on the left side of this table here, ipsilateral, so that's where the injection happened. You'll see that shows the pathology in the preformed fibrils, but also the contralateral hemisphere. So indicating that within the 30 days after this injection, that the phosphoserine 129 pathology is actually distributing throughout the brain away from the injection site. So it's not just occurring where these fibrils were injected. So diving a little bit deeper into this, so these are immunohistochemistry experiments, looking a little bit closer than just the overall. And what you'll see is so these now we're doing single bilateral intrastriatal injections, and what you'll see is monomer over 120 days isn't showing any phosphoserine 129 pathology, but preformed fibrils, especially after 60 days and 120 days, there's a lot of phosphorylated serine 129 there over time. And then this is where the injection happened in the striatum. Looking at the parietal cortex, which is distal from where the injection site is, you'll see again monomer over 120 days. We're not seeing any dark signal for phosphoserine 129. And I should indicate that the antibody used here is phosphoserine 129 specific, so it won't show a signal if there's no phosphoserine there. It doesn't just non-specifically bind to alpha-synuclein, it only binds to this phosphorylation site. And these preformed fibrils, again, were distal from the injection site. We're seeing pathology spreading here over 120 days. Looking at the substantia nigra, so this is a pretty critical region in Parkinson's disease, where it's a highly dopaminergic region, and these dopaminergic neurons in here die off in Parkinson's disease. So it is really a critical area to be looking at. And you'll see again, monomer is not inducing phosphoserine 129 pathology over 120 days, but preformed fibril is. So I think that's pretty strong evidence that these fibrils are initiating this pathology here. Looking at actual dopamine levels, so how is brain dopamine affected by these fibrils, you'll see monomer over 120 days here, and we're looking at the dopamine nanograms per milligram of total protein determined by LCMS, so using mass spec. And you'll see monomer over time, there's no change in the levels of dopamine. But in the preformed fibrils, especially after 120 days, there is quite a significant reduction in the amount of dopamine around. And we also did this mass spec with dopamine metabolites and found the same effect. So moving on to a rat model, we essentially find the same thing. So looking at the phosphoserine 129 pathology, again, monomer is not inducing anything over 120 days. Preformed fibrils, you're getting a pretty strong presence of phosphoserine 129 in the striatum where the injection is. Again, looking at the parietal cortex, so distal to the injection site, we're not getting anything with monomer, but these preformed fibrils, again, are strongly inducing phosphoserine 129 pathology. And the same thing in the substantia nigra. So preformed fibrils, pretty strong response, even after 30 days post-injection in these highly dopaminergic neurons. So again, same, looking at dopamine, the LCMS shows that there's quite a significant reduction even after 60 days in a rat model of dopamine production. And again, similar trend observed for dopamine metabolites. So these are images from Dr. Rihanna Leek at Duquesne University. And this is actually showing one of these other tools that we've developed. And that is an ADO-dye conjugated fibril. So we have several wavelengths of these available. We're looking at ADO 647. And this is basically showing in actual primary rat hippocampal neurons that they appear to be uptaking these fibrils. So the fibrils or some piece of them is being uptaken by these rat neurons. So here, MAP2 is showing, it's basically a pan-neuronal marker, so it's showing where the neuron outlines are. And what you'll see so that's in red, and in sort of gray here is the ADO 647 dye, which is associated with, directly covalently linked to the fibrils. And you'll see it's sort of concentrated within the neurons there, indicating potential uptake there. And what we really want to look at next is if we can actually start to see transmission from neuron to neuron of these fibrils. Okay, so I'll just remind you, again, we have this monomeric state and sort of the healthy cells, these oligomers that form in these fibrils. The fibrils are very interesting in terms of their seeding and their visibility in both Alzheimer's and Parkinson's plaques, but these oligomers are actually thought to be the more toxic structure. So we have been looking at those as well more recently, and we have successfully generated alpha-synuclein oligomers in vitro. So the first ones we were able to make are these dopamine-induced oligomers. So you'll see these are globular structures, 20 to 30 nanometers if they're not clumped together here on the left under EM. And the dopamine is physiologically relevant because when you think of Parkinson's disease and the death of dopaminergic neurons, these are neurons that have a high concentration of dopamine, and alpha-synuclein is working in these synaptic vesicles which contain dopamine, so it's not a huge stretch to think that there may be some role of dopamine here. And we are able to induce these oligomers with the presence of dopamine. And actually just recently, we've developed these native oligomers with no inducer, and we're pretty excited to start working with those, and they're an excellent control for the dopamine-induced oligomers as well. So here's our initial data from the dopamine oligomers, and we have shown that they do seem to be more neurotoxic than the preformed fibrils. So looking at primary mouse cortical neuron viability, and we're measuring this by mitochondrial dehydrogenase MTT activity. Basically, if the neurons are healthy, their mitochondria are healthy, they're able to degrade basically this yellow dye into this purple crystal, and you can read that out in an assay. And what you'll see here on the left, so that's the percent to vehicle control. So the vehicle control here is up at 100%. And you'll see several of the different types of fibrils here. And they do neuronal viability by about 25% to a significant degree. Monomer on its own is not affecting the viability of these neurons. And then on the right here, you see the dopamine oligomers, and they are significantly more toxic, so less than 50% viability compared to the 75%-ish viability from the preformed fibrils. So we have found that they are indeed more toxic, and we are currently investigating the native oligomers just to confirm that the dopamine isn't having any effect itself. So that was it for alpha-synuclein. I also want to talk about tau a little bit here. So tau functions to stabilize microtubules. So microtubules are pretty negatively charged. Tau has a high PI. It's more positively charged. It stabilizes these microtubules. And through some mechanism, and many different have been studied, when tau is not bound to these microtubules, and again, we're talking about a very flexible, relatively unstructured protein in solution, it also, similar to alpha-synuclein, starts to oligomerize, and then it eventually forms these paired helical filaments and these neurofibrillary tangles present in the plaques in Alzheimer's disease. Now, tau is normally regulated through phosphorylation, so the more phosphorylated it is, the less it's going to bind to the negatively charged microtubules. It's a regulatory mechanism, but probably the best studied mechanism is this hyperphosphorylation of tau that occurs, drives it away from microtubules, and then really facilitates this oligomerization and eventual sort of fibril filament formation here. And in Alzheimer's and Parkinson's, this hyperphosphorylated tau is present, and that's sort of a hallmark of these diseases. So we have been able to generate several isoforms of the tau preformed fibrils, several relevant mutations, clinical mutations related to Alzheimer's disease, several different sizes. So 2N4R is the full-length tau isoform. K18 is a more truncated fragment, as is DGAE. And recently, we've also been able to make the wild-type full-length tau fibrils. And in general, because we're working with recombinant E. coli material and there's no phosphorylation, a scaffold such as heparin is needed to induce large-scale aggregation of these larger constructs. And also recently, we've been able to generate this baculovirus SF9 expressed tau, which we've confirmed with mass spec that has over 30 sites of phosphorylation, so hyperphosphorylated, one could say. And these will actually aggregate on their own. So the phosphorylation is really driving this self-aggregation without a scaffold. So these don't actually need a heparin inducer. So here you'll see the THT seeding assay looking at relative fluorescence units over time. You can see the monomer on its own is actually forming fibrils, and also is seeded. So the higher green line there. Yeah, so it's pretty cool that they form on their own without a heparin inducer, and we're really excited to test these out in some mouse models. So far we've tried the tau K18 preformed fibrils injected into mouse hippocampus, and now we're looking at, and again with an antibody, tau serine 202, threonine 205, which has been shown to drive tau aggregation. And what you'll see here after injection of these fibrils into hippocampus is these tangle-like inclusions appear here, and the inset here is the negative control, so it's nice and clear. There's no aggregate detected by antibody observed there, and in the other boxes here you can see that sort of those tangle-like inclusions, and this is 9 weeks post-injection. So we are initiating a sort of Alzheimer's-like pathology here. And again, these are not phosphorylated before they're injected into the mouse model. So we also have generated tau oligomers, and this is pretty recent data we have, and using arachidonic acid. So arachidonic acid is a fatty acid, and it plays key signaling roles in neurons, and it's actually the second most abundant fatty acid in the brain. So basically, in the presence of arachidonic acid, tau will form these nice globular oligomers here on the left in a short incubation. And what you'll actually notice if you look on the right here, not only are there more oligomers around, but they start to elongate even. And these are all still soluble. So will these proceed to sort of a protofilament and a perihelical filament eventually, possibly? But we're pretty excited to test out these new tau oligomers in cell cultures and mouse models to look at their toxicity relative to preformed fibrils and pathology induction. Finally, we also have started working with a beta peptide. So it's a key component in neurodegenerative diseases, both Parkinson's and Alzheimer's. And essentially what happens here is you have this transmembrane protein that is cleaved by various enzymes to release this small soluble peptide that very quickly aggregates and forms these beta amyloid plaques, which are present in both Alzheimer's and Parkinson's fibril deposits. And this is normally in the synapses of neurons. This is occurring, and so it's relevant whether this is occurring before Alzheimer or before tau or alpha-synuclein and inducing that's definitely a possibility. But I think at the end of the day, it's going to be a combination of factors. So it's important to look at all of these different proteins and their oligomers and fibrils on their own. So we have recently generated these A-beta 1 to 42, so those are the amino acids, fibrils from a synthetic peptide. So what you'll see here on the left is a STS page Western blot with a 6010 antibody that will detect any A-beta. And looking at the gel here, monomer, you just get this nice clean signal for monomeric protein down there at 5 kilodaltons. It's 4.5. And then in the fibrils, you actually see on the gel here this formation in the well and at the very top of the gel here, so indicating a very large size. These fibrils are forming, and on the right you'll see sort of 3 different types that we have in development, and we're continuing to characterize these and are looking forward to testing these in models as well. Finally, we've also been able to generate A-beta oligomers from that synthetic peptide. So again, here on the left, STS page Western blot with the 6010 antibody. You'll see so the molecular weight ladder is on the very left lane, the second lane over. You'll see this sort of faint smear of 37 kilodaltons up, indicating that oligomers are actually forming here. And more obvious is the formation of these dimers and trimers around 15 kilodaltons on this gel here. And on the right, you'll see those nice squabular oligomers forming. So we're pretty excited to test out the toxicity of these A-beta constructs as well. So key takeaways overall, alpha-synuclein and tau fibrils generated in vitro will seed additional fibrils in vitro, as well as induce hallmarks of Parkinson's and Alzheimer's pathology in both mouse and rat models. So they represent pretty cool tools to study the disease initiation and progression in these models. Alpha-synuclein oligomers, which we generate in vitro, have an increased neurotoxicity compared to fibrils. And we're also really excited about the tau and A-beta oligomers that we've developed and are now looking into. So our key next steps here are we would love to demonstrate neuron-to-neuron transmission of these in vitro aggregates in a model to actually show the sort of spreading of pathology that we think is occurring based on the phosphoserine being detected in all different parts of the brain away from the injection site. And then we also want to fully characterize toxicity and pathology of our different oligomers relative to the fibrils. So I'd like to acknowledge Ariel, our president and CEO, for really organizing and finding funding for this work. Kristen has been a lab tech working on R&D of these new oligomers, as well as Vicky, Sophie, Kenji, and Alex for all the work they've done on the fibrils, and the ADO dye-conjugated preformed fibrils were done by Kenji. So I'm happy to take any questions regarding this talk directly to my email. I wish it was in person and I could answer in person, but that's the times that we live in right now. So with that, I'll just leave you with our last slide here if you're looking for any more information on our fibrils or oligomers, which are coming up really soon. For alpha-synuclein, tau, A-beta, SOD1, and TTR, there's a whole bunch of other proteins we're also working on. Yeah, so that's it. Thank you for listening.